Today I'd like to give you a more technical view on the cell-based assays we're producing to detect those autoantibodies, not only in the context of neuronal autoimmune disease, but also other diseases. And that will be the aspect of my short 20-minute presentation. So if we go a step back for a moment and look at the discovery rate of those autoantibodies, it all started with the discovery of Hansen and colleagues, which described an encephalomyelitis with a carcinoma. And that was really the first case, or a group of cases, where that provided like a stereotype of the diseases. But it took another 10 to 15 years, so in the 1980s and 1990s, to describe other autoantibodies that we now know as the classical paraneoplastic antibodies. Typical for those antibodies, and that's what you have heard already, it's clinical syndromes like limbic encephalitis, cerebellar degeneration, and almost always you find an underlying cancer. That has, of course, consequences in many cases. Immunoresponsiveness is quite low, so patients often have a poor outcome and ultimately die from these kind of conditions. So classically, if we try to describe those autoantibodies, of course, we're coming from a clinical picture. Here you can see this publication here by Joseph Dalmau. It's an iconic publication by now, and you already have seen it, I believe. So here, the group of Dalmau describes a paraneoplastic encephalitis with psychiatric symptoms, hypoventilation, or varioteratoma. So this is really now the stereotype for NMDA receptor encephalitis. Of course, these were mainly female patients, or almost exclusively female patients in this patient. So important is from the classical approach starts, of course, with the clinical picture and the group of patients that sort of share the clinical symptoms. And then afterwards, the target antigens are identified, and I choose this as an example here because I think this is a kind of beautiful picture. So here you see staining of NMDA receptor antibodies or binding to the rat hippocampus, and you see here this staining here, it's referred in the literature as the neuropil staining, and this is actually the location where the antibodies obviously bind. There are certain other assays, so you can test it also with living neurons, and also, of course, the cell-based assays the presentation is more or less about. Excuse me. So if we talk about how to find antibodies in the laboratory context, there are different methods out there, all of which have been applied to discover certain autoantibodies. So there's Western blot technology, often combined with mass spec. Then we have the classical radioimmunoprecipitation approach. Protein libraries can be used. Also radiolabeled toxins, so bungora toxin, dendrotoxin, snake venoms with an isotope that bind to specific ion channels like the VGKC or also acetylcholine receptor. Expression libraries can be used, peptide libraries, but all of which have certain drawbacks, obviously, and the main disadvantage to my point of view is that many of them are like a black box principle. So you dump something in and you get something out, and you don't really know what is happening in between. Many of those technologies rely also on denaturing steps, so denaturing agents that modify the antigens to a certain degree. So hence the outcomes often equivocal, and if we talk about huge protein, peptide, or expression libraries, even statistically unfavorable. So it's a really hard challenge for a company that try to produce no assays. EUROIMMUN itself has come up with a kind of unique way to describe and discover new antigens. It's referred as the UFO project at EUROIMMUN, and UFO stands for unknown fluorescence object. So what it means is that we see binding to here neuronal tissue that causes a specific staining pattern. So we know already there's an autoantibody that specifically binds to certain structures in the brain. What we do next is we can take those immunocomplexes, we lyse the tissue from the slides, we pull down the immunocomplexes with paramagnetic beads, separate them on a gel, and then subject them to mass spec in order to identify the proteins behind those immunocomplexes. And we have applied this quite successfully for many autoantibodies, actually. This is one example, the discovery or identification of flutalin 1 and 2 as the target of autoantibodies in a subset of patients with multiple sclerosis. So we sort of take the opposite approach. We use our technology to find antibodies, and then we go back to patient instead of the classical approach, starting with the patient. So you have seen a similar graph already. Neuroimmunology is really a rapidly evolving field. I think you have seen the identical graph actually in Patrick's presentation. So we're now at a stage where we can determine over 50 clinically relevant autoantibodies. And of course, this is also exemplified by the number of annual publication that are available through PubMed, which is getting more and more as we speak. The entire field mainly was revived actually by the discovery of aquaporin-4, an NMDA receptor you already have heard about. As the general concept, not only for the detection of those antibodies in the diagnostic workup, but also in terms of assay design, we have to distinguish two different classes. So on the one side, we have those intracellular antigens, which comprises of the classical autoantibodies or autoantigens like HU, YO, RE, CV2, and others. And then the emerging class of new antibodies, which mainly comprises of cell surface or synaptic proteins, so ion channels, receptors, and others. And this is the class where also NMDA receptor and other autoantigens fall into. And with that, of course, we have different challenges in terms of how to come up with a different assay and its design. So the first challenge actually is that often if we have an autoantigen on the surface of a cell, it often comes with a certain conformation. Those conformations are fragile. So during the process of purification, we actually have to make sure that the 3D structure of the protein is well maintained during this process, because once we destroy it, so we get linear structure due to denaturation of different origin, the antibodies in the patient sample will not recognize these epitopes anymore, obviously. The second challenge is that many of, or all of those antibodies, if we refer to the class of cell surface antibodies, obviously, are membrane-bound. And membrane-bound proteins is a real challenge in laboratory workup. So if you talk to biochemists, which are doing that like routinely in the lab, they will let you know that they don't want to do it because it's too complicated. Why is that? So the connection, of course, of many proteins is quite stable. So often they have transmembrane sort of domains which span the membrane multiple times. This results in low yield contamination. So purity is a problem because you often get parts of the plasma membrane into your protein fractions. Also, detergents obviously are necessary for isolation, and that might result in the destruction of the epitopes. So as soon as you start to purify this kind of proteins, you need kind of harsh conditions to purify them, and this of course reflects back then to the first point. So just to say it again, the problem here is the purification of a functional antigen. And from a perspective of an in vitro diagnostic company, this is really unfavorable because it's technically demanding. The purification steps are quite demanding. So it causes a quite lengthy development process with an uncertain outcome. So that's not a project you want to start with. So how to omit this? So the solution here for us was actually quite simple. So we omit the purification step. So what does that mean? That means that we use directly antigen-producing cells that are fixed. So the cells itself are the sort of the production facilities to provide us with the antigen. So this is a quite gentle and fast process, obviously. The assay format here is referred as recombinant indirect immunofluorescence, also known as cell-based assay, and also brings, of course, the advantage of multiparametric testing. So you have seen a similar slide before. What is a cell-based assay? Just in brief, we usually use a certain cell line. Usually those are human cell line, human embryonic kidney cells, for instance, but also others are used in the literature. We can introduce the DNA into these cells, and as soon as we do that, the cells are making the antigen of choice. Often those are receptor proteins, obviously, so the cell itself transports those receptors to the cell surface. Once that's done, you can also add the patient sample, either serum or CSF, If antibodies are in the sample, those antibodies will recognize these epitopes on the surface of the cell, and obviously this can be visualized by a secondary antibody, which is fluorescently labeled in this case, either by microscopy or other methods. So quite easy and straightforward technology. How does it look like? Just to give you an example, so this is the autoimmune encephalitis mosaic 6 you have just seen, and here you see a positive reaction, or a strong positive reaction of NMDA receptor transfected cells. Another important step is if we think about those assays, is that in terms of in order to provide you with a standardized assay, also a viable assay, to be honest, we had to add some fixation, because otherwise we would add living cells to the slide. We can't sort of ship that throughout the world, and also those cells wouldn't be stable. So what we have to do is we need to use some chemical agents in order to fix, to stabilize the cells on the surface. We have different protocols, and you also find different protocols in the literature. So whatever reference institution you're going to, they have their own little nitty-gritty protocol to do this kind of, or to make this kind of assays. One of those, or many of those protocols is based on formalin. So what does formalin result with when you treat HEK cells with it? So it really only more or less fixes the cells and leaves the cell surface antigen often intact. So this is a highly specific method. Surface structures are preferred in these cases, but it bears the risk that the epitopes might be altered. So there has to be per se a different fixation method. So the other protocol which is often used is acetone-based protocols. Acetone fixation has the drawback that it permealizes the cell membrane, so it gives other antibodies that are present in the same patient sample access to these epitopes. So mainly ANAs, for instance, can also be observed in these kind of cases. But it also is required for intercellular antigens, of course, but more importantly for formalin-sensitive epitopes. So for those antigens, formalin is not working quite well. We usually rely on the acetone fixation with a drawback of increased background binding. So how does it look like? Again, two examples. The first you have seen, this is NMDA receptor, and you can see this greenish background here, and then on the opposite we have aquaporin 4, which relies on a formalin-based fixation protocol where background is largely absent. And just to say it again, we need to do that in order to provide a standardized assay. Just one thing to add, because it causes some confusion. So here, of course, it's quite easy to see that it's a positive reaction, but sometimes you also have weaker positivity. So it's kind of hard to discriminate a real signal, or can be hard to discriminate a real signal from background. So what you have to know for this is each and every individual cell-based assay comes with a unique staining pattern. So they all have their own sort of footprint, if you want to say it like that. For NMDA receptor, a positive reaction on the NMDA transfected cells is usually observed by a granular cytoplasmatic staining that you can see here. And then those classical, more or less classical, cytoplasmatic extensions. So it looks like paint drops on a canvas, you know? So really, really specific. And if you look at the transfected cells, with this kind of knowledge, you're having a much easier time to recognize these kind of positive reaction, especially if we have high background reactivities. And just to say it again, each and every individual cell-based assay comes with a unique pattern. So once you're aware of those, you will find yourself having a quite easy time to recognize them. Obviously, not everyone is aware of this and hasn't been to the EUROIMMUN Academy to get like the proper reading skills. So this I found in a more or less recent publication where the authors actually tried to make a case. So what they did, they incubated EUROIMMUN slides with a patient serum, and here you can see the staining pattern. And from what you've learned just a minute ago, you already know this is not as typical staining, and they tried to make a point out of it by counter-staining, because now they said, okay, we added a specific antibody for NMDA receptor and it doesn't stain the same cells. Yeah, well, your fault. Proper training would be a good cure, actually. Another difference between the assays you find, especially in the context of research, is the differentiation between fixed cell-based assays, so also the commercial assays, and live cell-based assays. So what does it mean? Obviously, live implies that we're dealing with living cells. So the only mere difference is that the time point of when the fixation happens. So in the classical fixed cell-based assay, we prepare the slides. Cells are transfected with the recombinant antigen. Antigen is expressed. Then the slides are fixed. And after the fixation process, we incubate the sample with the slides with the sample, and then of course it can be visualized. In the live cell assay, usually living cells are incubated with the patient samples, and then afterwards these these cells are fixed with a fixative. Those two examples are from different research groups. So this assay here, the fixed assay, is used by the Dalmau group, and the live assay is still used by the Oxford group. And both assays try to detect NMDA receptor antibodies, and of course they do, but here is a clear lack of international standardization, and that is also something EUROIMMUN tries to push forward. So we are heavily involved in multicentric comparison studies. The best example really is Aquaporin 4. I think now so this publication here is the third round of this kind of multicentric studies. Here in this case, over 13 European centers are involved in order to help and improve the diagnostics with these kind of tests in the context here of neuronal myelitis optica. Similarly, we're also involved in the standardization process of mock transfected cells. It's essentially the same community, obviously, as the people from the neuronal myelitis optica field. And these researchers are really open to do this kind of studies, and we're always looking for partners to conduct these kind of multicentric studies in order to prove the diagnostics, the diagnostic tools, and the diagnostic accuracy of those tests that are out there. What else can we do? So also those neuronal cell-based assays are available obviously for our EUROPattern system, which is a automatic microscope system. And now with the release of the second version of the software, not only automatic image acquisition is possible, but also automatic classification. So the software tells you or discriminates positive from negative cells and helps also to bring in more diagnostic accuracy. Obviously, cell-based assays are not only available in the context of neurology, but also for other indications. So there are tests for skin antigens, for instance, involved in bullous dermatosis. Other antigens are associated with autoimmune nephropathies, so things like PLA2R, THSD7A, and also in the context of Crohn's disease and all sorts of colitis, celiac disease, also infectious diagnostics, autoimmune liver disease, and myositis. For all those indications, we can supply cell-based assays which offer certain benefits in different contexts. And last but not least, I want to go back to the neurology portfolio. Neurology really is an increase in area of interest, not only outside of EUROIMMUN, but certainly within EUROIMMUN, and was identified as one of those big synergy projects, also with new partners, not only in EUROIMMUN, but also outside of EUROIMMUN. And we try to constantly expand our portfolio, not only in the range of autoimmunity, but also in the range of neurodegenerative diseases, where we're quite active and can provide a quite unique portfolio. And with this, I come to an end, and thank you for your attention. Thank you very much.